Welcome and thanks for joining us today. My name is Matt Willard and I'm the Senior Marketing Manager here at Fluence Analytics. Before we get started, I want to point out the chat feature at the bottom of your screens. You can use that feature to make comments during the webinar or to pose questions to our panelists, which they will answer at the end of the webinar. Fluence Analytics is a startup headquartered in New Orleans, Louisiana, that spun out of Tulane University in 2013. We manufacture industrial and laboratory monitoring solutions that produce continuous data streams and enable real-time optimization. This leads to improved process control and faster R&D periods. Today's webinar will discuss ACOMP, Fluence Analytics' patented solution for automatic continuous online monitoring of polymerization reactions. Our panelists are Dr. Sig Floyd and Mike Drinsky. Dr. Floyd is the Business Development Advisor here at Fluence Analytics, and he is a global chemicals and equipment manufacturing executive with 30 years of experience at various international companies based in the US, Japan, China, and France. Mike Drinsky is the CTO of Fluence Analytics, and he has more than 15 years experience developing ACOMP's hardware, data analytics, and proprietary technology. Again, thank you for joining us today. And at this time, I'll turn the webinar over to Dr. Floyd.
Thanks, Matt. Once again, good morning, everybody. It's Sig Floyd, as introduced, and today we will be talking about ACOMP, kind of a mouthful of a word, so let me just say right away, it stands for Automatic Continuous Online Monitoring of Polymerizations. That's our system. I will be talking mainly about the commercial value aspects of using ACOMP. I'll just say a little bit about how it works, and then about halfway through the presentation, I will hand off to our CTO, Mike Drinsky, who will cover in much more detail the technical aspects of the system and also introduce a case study. So our company, Fluence Analytics, is a very new company. It's a startup that was spun out of Tulane University in New Orleans in 2013. But the ACOMP itself has been around for some time. It was invented a little more than 20 years ago, in fact, by a professor of physics at Tulane University, Professor Wayne Reed. And today he is our chief science officer, so still very close involved with the company. So our profile, I've just mentioned a little bit. We do have the IP or the patents that were originally mostly developed in the Tulane period. Those have been obviously contributed to the company. We are in New Orleans, very close to Tulane University, just a few miles away. Our facility is about 6,700 square feet if you're using the English units. And we have here a team of nearly 20 people here working on all aspects of commercializing this technology. Just mentioned, because it's not on this slide, but we also received a Series A funding round in May of this year, so also confirmed strong investor interest in this technology. And really our mission is to combine a hardware system with advanced software and data monitoring, data processing, analysis in order to provide solutions particularly for the chemical and biopharma industries. So our products are intended to help you with productivity. I'll just mention we have two main product lines, ACOMP being one of them. The other one we're not going to spend any time on today, except just to mention it right now. It's called RGen, which stands for Aggregation Rate Generator, and it's a laboratory instrument that has 16 independently controllable cells. It uses light scattering to look at things like protein aggregation, or it could look at degradation of polymers, other natural products. And this product is mainly targeted at the biopharma industry. So just a little footnote here to say that's one of our product lines. The product line we are discussing today, the ACOMP, actually comes in two variants, although the content is very similar. The industrial ACOMP there in the middle, the IACOMP, of course, is for industrial use in the petrochemical or chemical industry, specifically for monitoring polymerization. The lab ACOMP, on the other hand, is intended for use in an R&D environment where it can increase productivity of R&D. And so we'll talk a little bit more about that later on. So the main problem faced by the polymer manufacturing industry is really one of understanding what is happening, what is going on in the reactor, what you are making. As a matter of fact, I started my career more than 30 years ago now, and my first job out of school, I was involved in the rubber industry, and we would be running a measurement called the Mooney viscosity. So the way it worked is you would take your sample out of the reactor, run that over to the lab. You would then have the lab generate the measurement after a certain point, run that back over to the control room. And so, of course, this is a fairly inefficient way of really understanding what is happening. As it turns out, we are now in 2017, almost 18. Actually, nothing has really changed in this picture since that time, and the Mooney measurement is run exactly this way by the rubber industry manufacturing polymer. So, you know, this is the inherent inefficiency that is very common in polymer manufacturing. So what does ACOMP bring to that? Well, ACOMP, first it is, point out, must be directly connected to the reactor, as we show here. And when that is done, using the internals of the ACOMP, which basically the sampling system, detectors, sensors, and then software, we are able to generate continuously, automatically, as the name says, and in very close to real time, raw data, and the data is then processed using our internal algorithms, which are quite sophisticated, and that data can be supplied directly back to the plant control room, the parameters of interest, whether those be a residual monomer measurement, for example, or a viscosity measurement. This can be fed back directly into the distributed control system in the plant. So that entire process gives you greatly improved control or understanding of what's going on in the reactor, which can then be used to optimize your control. So the way I often like to talk about it is polymerization reactors are basically a black box, but when you have the ACOMP system, you have a window inside the reactor, and you can actually begin to understand what is happening there. So I'll just do the sort of the very basic version of how it works, because Mike will come to much more detail on this. ACOMP is, as I said, connected to the reactor, and we continuously extract, dilute, and condition a stream directly from the reactor, and then we pass it through the detectors at a very low concentration. So we dilute that stream generally by a factor of 100 to 1,000 to a low concentration. Why do we do that? Because our detectors are all relying on the properties of polymers in terms of their interaction with light sources. So whether UV or laser light or different types of light. So therefore, we need to have the polymers in a mode where they are fully diluted, not interacting with each other and not interacting with anything else that might disturb the signal. So we're really looking at very intrinsic properties of the polymer. The typical extraction rate from the reactor itself is very small, of the order of a milliliter per minute, so very small quantity. There is some delay time inherent in the ACOMP because we have piping and tubing that extends from the reactor to the ACOMP and then within the ACOMP a little bit too. So there is an inherent delay time there, but compared to the time scale for sending things to the lab, which is minimum, let's say, 45 or 60 minutes and sometimes can be many hours, the delay that is inherent here in the ACOMP is only of the order of 1 minute, maybe up to 5 minutes. So this is essentially real-time measurement. We also want to point out, there may be many of you in the audience have a lot of experience with chromatography, so we want to say up front that ACOMP is not a chromatographic process. We are not putting the stream of liquid from the reactor through columns. We are putting it through the detectors directly, continuously, as I mentioned. So this is a definite difference between ACOMP and typical size exclusion chromatography. On the other hand, if we wish, we could put GPC or some variant on the end of the ACOMP system. The actual stream that is coming out of the exit of the ACOMP is very suitable for GPC analysis, so that is possible to do, but it is not part of our standard offering. And actually, just a little bit of nomenclature, what we refer to as the front end in the ACOMP is the first part where we are extracting, diluting, and conditioning that sample, and then we call the detector train part the back end. We have 4 standard detectors, which are UV absorption and viscosity, and then differential refractive index. And our most sophisticated detector is actually the static light scattering that was, has been developed and improved by Professor Reed at Tulane and Mike Drinsky and many people. So that is our most sophisticated detector of the 4. And we can add other detectors optionally. We could add GPC, as I mentioned, but we could also add other things depending on the requirements of the customer and what you're looking for. So coming out of those detectors, again, we will get raw data, and Mike will explain that in a few minutes. And then the raw data is in turn converted by both physical theory and our algorithms into properties of interest, such as molecular weight, the intrinsic viscosity, which is sort of a zero concentration limit of viscosity, composition, monomer conversion, and residual monomer. What's very important too about the ACOMP system, and one of its, I think, unique advantages is that it is able to track process upsets or disturbances in real time. So this is a feature that allows for intervention when things are not going right. Where we see the value of the ACOMP system, basically we like to break it down into 4 categories. We've actually done a lot of quite detailed work to quantify what we're going to talk about here, but basically the benefits, of course, cost. If it's a batch process, we are typically able to reduce cycle times, and we have a case study to show you exactly how that's done. With continuous process, it's about faster grade changeovers. From efficiency standpoint, of course, we are going to have less rework in the system, reduced manual sampling, really a lot less analysis is going to be needed if you have the ACOMP running. The data is fed seamlessly into the plant DCS system where it can be used to implement control algorithms directly. So those are benefits. And then quality, likewise, by being able to control things like the cycle time or the transitions, you can envision having tighter specifications. Of course, you reduce the amount of off-spec product. In batch, we'll show you an example later where we basically avoid degradation when you run the batch reaction too long. And then last but not least, for health, safety, and environment, we of course reduce manual intervention, so the operators do not have to be exposed to hazards in running back and forth collecting samples of reactor liquid. But overall, for a site, you can envision that using this type of analytical technology, you can reduce the energy footprint, you can reduce the materials consumed, and so there are significant HSE benefits as well. So we, in terms of return on investment, we are going to make what will sound like a quite bold statement here. We believe that using this type of technology, you can quite easily, in fact, generate returns of over 100% return on investment, annually, meaning that the payback, is less than 1 year, typically. And why we're able to say that, just to talk about two generic examples here. If it's a batch process or semi-batch, and let's say you have a 20,000 ton plant with a $50 million turnover, the major benefit, to focus on the major benefits, is going to be cycle time reduction, and we can say that it's possible to get the order of $1 million. And how do we arrive at that? Basically, if you have a 5% reduction in the batch cycle time and you're able to sell that capacity, so it depends on the economic environment you're in, but if you're able to use that capacity, $50 million times 5% is $2.5 million, and then the cash margin, if the cash margin is 40%, that is how we get to that $1 million figure for the batch example. Very, very straightforward. As I'll mention later, the batch cycle time can be improved by significantly greater than 5% in some cases. For continuous process, likewise, let's say you're running a plant and the annual turnover is around $150 million. Again, your big nugget there is going to be reducing the transition time where you are producing what's called often twilight material, or basically off-spec grade in between two grade specifications. So if you can reduce that transition time basically by just 1%, you are looking at of the order of $1 million there. Oftentimes that transition material is sold for less than half the prime price. Sometimes in the rubber industry, it would go into shoe soles or something that's a very low value application. So by reducing that transition time, you are able to generate very, very significant savings just on that parameter. So how about the actual industrial performance of the ACOMP system? ACOMP has been in business now for close to 3 years. Our reference example that we're going to share refers to a customer here in Louisiana. It's a global specialty chemical company. It's making a polymer that is frequently used for water treatment. And we have, with the Generation 1 ACOMP, close to 3 years now of experience with 98% or really close to full on-stream availability, meaning whenever the customer is using it, the equipment is available. I mean, if they're down for cleaning or something like that, we don't count that time, obviously. Generation 2, so they were very happy with this. Payback was very fast. They bought another unit. This is the Gen 2, lower right corner there. And so they bought that unit, and that unit now has well over 1 year experience as well. And although I mentioned in the example where you can get very significant savings if you can just reduce batch cycle time by 5%, we have gone back and looked at a lot of the customers' data, which is shared with us, and we have estimated an average savings in cycle time of about 17%, so very, very significant. Now, the economic benefit of that saving is going to depend very significantly on what you do with that saved cycle time. In a very strong economic environment like we're in right now, you probably can sell all of that capacity. In a weaker economic environment, the savings would be reduced amounts of overtime, a little bit of reduced energy usage, and we have actually done estimates around those things. So we are only using the 5% number to actually claim the very significant benefits for ACOMP, but the savings can actually be significantly greater. So payback was less than 6 months in this particular case, and they also saw quality benefits through tighter process control, basically keeping the batch time uniform and shorter than the original batch time. And by the way, they are running something like between 15 and 20 grades, let's say, and every time they want to run a new grade, they will use the ACOMP to optimize that cycle time as well. So, I mean, we are coming to Generation 3. It's just been released. We had a press release just in past week, and we've announced several improvements, which perhaps Mike will get into a little bit later. So I'd like to, at this point, hand off to Mike to carry on the presentation. Thank you.
All right, thank you, Sig. As Sig said this, I'm Mike Dorensky, CTO of Fluence Analytics. What we're going to look at now is some case studies, as well as I'm going to go over and describe how the data is collected and what we end up doing with the data from the system. In this particular first case, we're looking at a batch process. This is a free radical terpolymerization of methyl methacrylate, butyl acrylate, styrene with a solvent of butyl acetate. The conditions are typically, pretty straightforward for this type of polymerization. But if we start when we look at the beginning of the reaction process, we have pure solvent going through the detectors. There's really low signals here, but when we start to add monomer to the process and see that in the detector streams, we can see that the refractive index detectors start to increase because of the differential refractive indexes change. The double carbon bonds from the UV absorption start to absorb in the detector, and we can see the signals increase accordingly. But what we don't see is the viscosity or the light scattering characterization going up because there's no molar mass being built up quite yet. So those signals don't quite show up here at that point. At this point in the polymerization, we kick it off by adding the initiator at temperature so that the polymerization process starts. And what we see is the signals change accordingly. In this case, you can see the purple where the UV starts to drop down. And this is a function of the double carbon bonds being cleaved open and polymerized into the polymer chains. The refractive index starts to go up as according to the index refraction of the polymer being higher than the monomer itself. And then you start to see the molar mass detectors, the viscosity and the light scattering signals increasing from there. So from the refractive index and the UV absorption, we can get the monomer concentration as it's being changed in time, as well as the polymer concentration, in some cases being just the mass balance differential. Knowing the polymer concentration and then the effects of the molar mass detectors, we can get the molecular weight from that directly. That's the weight average molecular weight and the intrinsic viscosity. Now, from the intrinsic viscosity, if you know the Markowitz equation and the parameters that go along with that, you can actually get intrinsic viscosity or molecular weight by viscosity as well. Now, let's look at a couple of case studies. First case study is, as Sig mentioned, we have a system here in Louisiana that's been running for a few years, and this is a little definition of how we end up with that 17% cycle time. So if we look at the raw signals on the left, we see the blue being the UV signal being interpreted into monomer concentration, in this case ppm. The red signal itself is the intrinsic viscosity or reduced viscosity, in this case, pointed out in grams, centimeters cubed per gram. Now, if we follow these signals from left to right, we see the solvent baseline. We go to the monomer baseline where the absorption of the monomer baseline is much higher. But then as we start to polymerize, we can see the blue signal dropping down. At the end of this polymerization, you can see the blue doesn't actually reach the pure solvent baseline, and that's because the polymer chains are so long that they actually start to scatter some light. So what we end up doing in this case is showing that following the kinetics of the process, we can actually fit a value to that kinetics and actually predict when they get to a ppm. And this is what the customer is actually interested in. They want to know when they reach a residual monomer target value so that they can dump this product and start a new batch. What they're doing historically is they're following a recipe. This recipe leads them to taking a sample at a specific time in the process, running that to the laboratory, waiting for an HPLC measurement to come back, and that could take anywhere between 30 minutes to an hour. So what we can do with this case is we can actually point out when they finish the process based off the kinetics of the process. And the savings time here, as you can see, is on the order of an hour, 2 hours in this particular case. But what you also see, as Sig was mentioning, is we look at the reduced viscosity on the top and the red signal there. Polymer degradation starts to take place. So we're beating this polymer up, they're recycling it, they're keeping it held at high temperature, attempting to burn off the monomer, where it's not necessary because they've already reached their product grade quality. So we can help them eliminate off-spec product and help them eliminate coming in with failed batches because they're not meeting specifications on the monomer residuals. So what this can give us is an increased cycle cycle time savings, so they can reduce the batch time so they can just produce more batches in time. The kinetics of the process, so they can start to modify the kinetics of the process so that they can increase the overall batch times themselves by increasing the ratios of the monomer to the initiator, producing the bright size products, and overall reducing the batch process. And then like I said, the improved product quality. So there's no point in degrading your product, waiting on the process when you know that it's finished. All right, so now the next case we're going to look at is a continuous process. This is a case study here where we prove the idea of monitoring the process in real time. So imagine we look at a continuous grade transition for a process where you're running from, say, grade A to grade B. Grade B being on the right side, the grade A being on the left side. Properties here, we're going to assume these are in Muni values. And as Sig mentioned, that's a very standard value in the synthetic rubber measurements. So some of the assumptions we'll take here, assuming they're taking a sample from the process every 30 minutes, running it over to the laboratory, and it takes 60 minutes to get a process value from that to tell what the Muni value is. So as you can see from the red data points, there's a significant delay in actually determining what the product properties were in the process as it was taking place. Now, what this leads to is the intermediate measurements are very poor for process control. This has been a known problem in the industry for long years, but they're working this way, and they see the value in actually continuous and online monitoring and characterization. So if we look at what would the case be with ACOMP, as Sig mentioned, our typical delay times are anywhere from about 5 minutes of delay time from the process to the detector stream with a calculated value. So in the blue line here, we can see that very little delay, very little difference between what the process is actually doing compared to what the ACOMP is monitoring. What this leads to is we know when we're in specification. So the green lines here on the right, we see that's the high and low values of the specification. And we can tell when we actually reach the specification, so there's no delays there. In this case, we point out there's at least a 2-hour delay based on individual laboratory measurements. So that's 2 hours of product that's just going out and being pumped into a different tank, potentially wasted or processed into a different grade of product. What this also leads to is knowing when you've lost the previous grade specification. So when you're in this transition period. So what this points out is that at this point we can stop collecting the previous grade A, take this intermediate batch product or the intermediate process product, put that in a holding tank, and then know exactly when we got to start collecting the second grade. And in this particular case, we're going to show some of the more sophisticated things that we can do with ACOMP. And this has actually been done in the laboratory environment at this point, but it's very exciting, novel work that we've been pushing for. So in this case, we're looking at a batch process. This is a polyacrylamide process where we're actually doing a free radical polymerization. And according to nature, free radical polymerization should start out with high molecular weight in the beginning. And as the monomer is consumed throughout the process, you get lower and lower, smaller and smaller molecular weight chains. So what we actually end up doing is knowing the molecular weight as it's being built up, we can adjust the monomer to initiator ratios so we can continuously build bigger and bigger molecule chains throughout the process. And that's basically against what nature wants it to do. So what we did is we took this blue line here. If you can see the model line, we say this is our trajectory model. And as we're monitoring the process in real time, we can adjust the molecular weight and intrinsic viscosity accordingly so that we follow this model value. What this gives us is, as I mentioned, the process that we use this characterization tool is that we can actually go counter to normal evolution of the batch process. So we actually build molecular weights up to what we want them to be, as opposed to what the kinetics, the reaction processes, and the true nature of the product actually wants to go towards. So we can actually make products based off of what we want them to be. All right, so now let's look at a couple of different versatility options for the ACOMP itself. We do have a lot of experience with batch, semi-batch, and some experience with continuous processes. And we've actually had quite a bit of experience on laboratory with solution, bulk monomer, emulsion, and inverse emulsion processes. And our industrial systems are actually working in a little bit of each of these spaces themselves. The chemistries that we've had quite a bit of experience with are anywhere ranging from acrylamides, acrylics, styrenics, urethanes, and things like that. So we do have extensive use of the system to characterize all these different types of properties from these different polymerization and chemistries. But the more exciting things that we're pushing toward these days are toward the polycarbonate, polyester, vinyl acetates, and silicones, because these processes have typically been a bit out of our reach at this point because of the high pressures, the high temperatures needed to run these polymerization processes. But we're moving that direction. We're actually having quite a bit of success moving that route. So now we'll look at a couple of different options. As Sig mentioned, we have a couple of different options for ACOMP, laboratory and industrial. Laboratory ACOMP, if you look at them on the surface, they don't look entirely different. They come in a big box, but the difference really is in interfacing capabilities of the system and what they're designed to do. Laboratory and pilot applications for ACOMP gives us all the same monitoring, concentration, conversion, molecular weight, and intrinsic viscosity values from the process. It can be tailored to a particular type of chemistry. Maybe this laboratory researcher wants to work on basically all styrenics. So the system can be tailored specifically for that. All the software, automation, control, monitoring, analysis, and procedures are basically similar to between the two systems because all the calculations, analytics, and everything come out the same. What we do have here is customized report generation, review, and recovery of the data so that on a scientific level, the scientists can actually extract the data, take that to a third-party software, and do a higher level analysis on the data that we might not be conducting in the ACOMP software itself. We can collaborate on that level to develop that analyses and then add that as a feature or package that comes along with the ACOMP itself. As detectors go, we typically use the viscometer, the light scattering units, the refractive index, and UV, but we have options for adding different detectors based on the chemistry, the applications, and the needs. And Sig pointed that out. We've had experience with polarimetry, conductivity, pH, and a few other different detectors. And as he also pointed out, that the sample stream coming from the ACOMP is already basically diluted and conditioned for GPC. So the sample stream coming out can be taken directly and then injected into GPC. And we've actually been working on some of this applications and improvements themselves so that we can tie GPC directly to ACOMP. But the best part about this is, as you're doing applications research, developing new chemistries, you can get a better, clearer picture about what's going on with the process. So unwanted phenomena, real-time process perturbations, runaway reactions, and off-spec products. So you get this in real time to find out, okay, if I follow this procedure, I'm going to get the right product every single time. So the main differences between the ACOMP that's designed for industrial space is it's climate controlled. It has an option for doing classification design, so that it can be done in class 1 div 2 environments for explosion proof. The hardware and automation are basically very similar on the internals, but it has the option to integrate with the distributive control system and a historian so that the data coming from the ACOMP goes to the plant's historian and is saved there. It also has the ability to do very specific characterization to the process itself. So the operators need to know when the batch is done, or the operators need to know when the viscosity measure meets this specific grade, so that the ACOMP tells the operators exactly what they need to know, as opposed to flooding them with a lot of information that doesn't need to be into the process. Overall, the detectors, again, could be suited out with all 4 detectors that we have available to us right now, but we would specify those down to the only ones that are needed for that process. So there's no point in adding extra detectors that are not needed. They don't give you any beneficial data from that process. And this, at the bottom there, we said this is ready for deployment across a lot of different chemistries and wide range of product processes and methods. So as the generations evolved over the years, we can take a look at a couple of different options here. So the first generation of hardware was really built on a joint development. We worked together with the customer to develop the system specifically for them. But the system itself has a lot of things built into it. Over 50 sensors monitoring the health of the system, pressures, temperatures, currents, regulators, and stuff like that. So we always know what the system's doing, how it's performing. If it's starting to fail, we can go out and make sure that it's fixed before actually anything happens to the system. Generation 2, we had smaller space constraints, so we made the system a little bit smaller. So reduction in footprint and size. The overall operator intervention, there's none now. So it's built so now it's tailored and slave to the process. So the operators that are running the process don't actually know the ACOMP's there. They're not doing anything. It just tells them when they're done. We've also added the option for remote sampling. So we can actually put the sampling point very close to the process. And the ACOMP cabinet itself, as you see here, can be up to about 10 meters away. And overall, improved modularity, the detectors, sensors, and things like that. Generation 3, the biggest advancement in this one is now we have temperature and climate control of the fluidics. So now we notice the solubility issues of some of these samples and polymers that are less soluble in the different solvents. So we have to keep the temperature raised. And this is very typical of GPC, raising the temperature up to about 45 to 60 degrees Celsius. So now we actually have the ability to control these temperatures within the ACOMP system to improve solubility, improve our accuracy and integrity of our values. We've also added upgraded detector sets, so we now have proprietary detectors that we developed over the years, remote user GUIs, so the user actually doesn't have to be near the system to actually operate and understand and control what's going on with it. And our new chemical database and structure of our software gives us the ability to build in recipes and then extract those over time as you need them. And overall, I just want to thank everyone for their time and interest in the ACOMP systems. And here's a few different pictures of the different systems. On the very left is the first system that we installed. And you can see in the process environment there next to the reactor. The one in the middle is the generation 2. We don't have a picture of that one. And then the one on the right is the system that we actually just installed at a laboratory environment a couple weeks ago. Thank you. We're going to open it up for questions, and Matt will be driving that.
Okay, thank you everyone for attending today's webinar. If you do have questions for either Dr. Sig Floyd or Mike Drinsky, you can use the chat feature and just send them our way, or you can use the question and answer button at the bottom of your screen. Our first question is for Dr. Floyd. What is the smaller reactor volume that ACOMP can be implemented on?
Yeah, actually, this is a joint question, but we can go down, to 1 liter or.
In the laboratory environment, we've actually monitored processes on the order of a few milliliters. It really depends on how much sample we need to pull from the reactor. And the scale ranges from anywhere from a few milliliters for the reaction process, that might be a continuous or a batch process, all the way up to thousands of gallons. So our industrial systems are monitoring processes on the order of 20,000 gallons themselves.
Okay. The next question, is it applicable to slurry phase, also to gas phase reactors?
At this point, no. All of our solutions all of the samples have to be in solution, but that is something we're working on. We actually have proprietary methods and IP behind that space, and we're working with getting grant funding from a few different sources to help us improve the capabilities of the system for those sampling needs.
Thanks, Mike. Next question. Could we associate directly or indirectly the signals we are receiving to reactor fouling or tendency to form agglomerates, process upsets? Yes, that's actually one of the novel uses of the system. Since you can start to see these process differences, say potentially fouling. If you're producing gels in the process, fouling would show up in the ACOMP system as different pressure differences, possibly changes in the reduced viscosity because the molecular weight increases, but your hydrodynamic volume doesn't increase because of cross-linking and gelling and stuff like that. So you start to see these signals peek out of the background, and that's some higher level analytics, analysis, and things like that
that might be consequences of something taking place in the reactor that you'd have to look at on the back end.
Thanks, Mike. Next question. I didn't see polyethylene or polypropylene on your list of polymerization reactions. Will it work for these processes?
It can potentially work, but as we've mentioned, it depends on the process type. Basically, polyethylene or polypropylene, especially polyethylene, there is a solution process for polyethylene. So we do feel that ACOMP can be applied to that process. For slurry, it's going to be a little harder. Basically, the way ACOMP works, it's essential to get the polymer back into solution, fully diluted, in order to run it through the detectors. So if you can get the polymer into solution, we can run it through ACOMP. Polyethylene and polypropylene, things like that, are definitely on our longer range roadmap. We do have some interest, certainly, for polyethylene, and we think that the first place that ACOMP will be applied there will be in the solution processes, and can also be used with metallocene technology.
Okay, next question. How long does it take to build and install a unit in a process?
We're looking at about a 6-month turnaround here. So the ACOMP system itself, we can build in about 3 months. The rate limiter is typically making the connections, the tie-ins to the process with the company. So making sure that everything's, the utilities, the tie-ins to the process, and all that stuff is settled before we figure out where the system's going to end up being installed.
And the next question. Please let us know about the solvents of choice for ACOMP.
That really depends on the solvent that's used for typical characterization of the process right now. So I could say for synthetic rubbers, it's usually some type of hexane. If it's an aqueous polymerization, we would use an aqueous solvent to do the characterization, and that really depends on typical characterization right now. Say you're taking a sample and running it through GPC, we would probably use the same solvent that you're using for GPC or HPLC measurements. In the higher level correlations where Sig was pointing out we were doing work with correlating to Mooney, we use the hexane to do our characterization in the ACOMP system itself to get intrinsic viscosity, reduced viscosity, potentially molecular weight as well. And then that correlation is built up with how the rheologic measurement of Mooney viscosity is made. So those are the couple of different solvents there. But overall, we would use typically what is used to dissolve the sample for any other characterization means.
All right, let me just add one comment to that, is that if the reaction solvent is used, or if it's benign, the flow exiting the ACOMP can be recycled back into the process. However, having said that, the waste stream generated by the ACOMP would be of the order of a drum per month. So it is not very large. Even despite the dilution, we are not generating a huge amount of solvent to throw off there.
Have you ever implemented the system to any particulate process, for example, polyolefin area, regardless of the scale?
No, I don't have any experience with that one. No, unfortunately, no, we don't have experience with that particular case.
What does one of these units cost?
Okay, so yeah, we were anticipating this question for sure. Because it is a webinar format, we would like to encourage you to come back to us to discuss this individually. The reason I don't want to throw out a number is because it can vary very significantly depending on the implementation mode of the ACOMP. Is it industrial? Is it lab? What chemistries will you be trying to handle more than one reactor, which, by the way, is something we can do. So all of those things have significant implications as to the cost of the system. So I really want to encourage the person who asked that question, but also any participant, to approach us separately on this point.
Can ACOMP be used in online grade optimization of grade transitions in solution polymerization process?
Yeah, that's actually very much what it's used for. The idea is, knowing when you're on the specific grade and then when you transfer or transition to the next grade, understanding the differences between those two grades. And that could be any characterization product value, say the viscosity, molecular weight, the weight average molecular weight. But the nice part is you're getting continuous values from the system. Every second you get a new data point. So there's no significant delay in understanding when there's any perturbation in the process to say you need to adjust your monomer flow, your initiator or catalyst or something like that.
Can someone come to your location to see a unit in operation?
Yeah, we do have laboratory units here. They're not going to look like our industrial or specific systems, but we do have those in manufacturing. So we're going to have a couple of systems here, very shortly that are being built and tested for QA and QC purposes. But if you wanted to do, see a system in action, running polymerization, collecting data, that we do have laboratory systems to do that.
Yeah, let me add to that too. It is in the press release that we had sent out about a week ago, but we have just announced also our demo program. So these units that are being built and are going to be tested will also be available for demo trials at the customer's site, although I have to add at a cost, which will be determined. But we are looking at basically placing demo units at several customers' sites right now. So if folks are interested in that, please again come back to us.
Will variations in polymer concentration cause issues with the ACOMP measurements?
Yeah, that's actually handled by the ACOMP instrument itself. So if this is a continuous process and we're monitoring the molecular weight or viscosity, we would typically get the polymer concentration from the mass balance of the products going in. If there's a detection means by using UV or refractive index, we can get the polymer concentration directly as it's going through the detectors. So then we have a direct correlation from polymer concentration to molecular weight and viscosity. But in processes like a batch process, you're obviously going to be changing polymer concentration from 0 to 100% throughout the process itself. So we monitor those in direct real time.
Okay. Thank you, everyone. That's all the questions that we have. I'll give Mike and Sig the opportunity to share some closing remarks with everyone.
Okay, well, basically we do want to say that ACOMP can deliver very significant value in many customers' processes, many different chemistries, also on the R&D side as well. So we welcome your feedback. Thank you very much for joining us, and welcome your feedback and further questions. Just please come back to us.
Yeah. And same for me. Thank you everyone for attending. I'm open to answering any questions by email or contact through a webinar or just a direct email or contact from that. So thanks again.
Okay. Thanks, Mike. Thanks, Dr. Floyd. As you received an email from me, I will share this webinar with everyone who registered, and I encourage you to share it with your colleagues and coworkers or anyone else that you know who may be interested in such a technology. So thank you again for joining us today, and enjoy the rest of your day.
Thanks very much.